Antimicrobial peptides hold a lot of promise as new anti-infective drugs. Other functions may include antiviral and anti-cancer effects, as well as possible roles in neurology. To discuss antimicrobial peptides with me today is Professor Jens Rolff from FU Berlin. Professor Rolff, it's great to have you here. Thank you for taking part in this webcast.
Thank you for the invitation.
Professor Rolff, you recently published a number of interesting articles on antimicrobial peptides, including a prominent paper in Science magazine. When it comes to antimicrobial peptides research, where have we made most progress in recent years?
Well, I guess that's quite a wide-ranging question. I would just like to highlight a few areas, and they're mostly areas that are more closer to my expertise as an evolutionary biologist. I think, from a pure science perspective, one thing which I find particularly interesting is that in the past we considered AMPs more like similar to broad spectrum drugs, but it turns out that they can be actually very specific to particularly specific bacterial infections, for example, and that just very minor changes of one amino acid make a huge change to their specificity or even to their activity. I think that's one important aspect. The other, from the kind of pure science perspective, I think, is work that has been done in an insect model, in a weevil that basically keeps mutualistic bacteria in its gut or in specialized organs, so-called bacteriocytes, and they express a specific antimicrobial peptide only there to produce their symbionts. So I think that's something which in the past has been a bit overlooked, the importance of antimicrobial peptides as an interface with the microbiota, and especially if they've got players in the microbiota that are highly co-evolved with their hosts, which is often indicated by having an eroded genome, so they couldn't live as free-living microbes anymore. They're really specialized. So that's kind of, I think, from the pure science perspective. I think another thing very close to my interest is also that over the last 4 or 5 years, it has become more generally accepted that we have a problem also with resistance evolution if we were to use antimicrobial peptides as drugs. Because in the past, in the literature, it was very commonplace to say, oh yeah, antimicrobial peptides, they target what Michael Zaslow called an Achilles heel of the microbes, and therefore resistance evolution is highly unlikely or completely impossible. And we can discuss that in more depth. We know now that clearly resistance can evolve against AMPs. It's much more difficult, but still it is an issue we need to consider. And I think in terms of insights from the applied side, I think there are two things which I think are important, is that it's very clear that antimicrobial peptides can be very efficient in attacking biofilms. Now, bacteria in the lab are usually studied in planktonic cultures, so liquid cultures, but in nature and also in infections and patients, the kind of predominant lifestyle of bacteria are biofilms, and they are notoriously hard to treat. And AMPs might hold some significant promise for that because they are able to attack non-growing cells. And that's what we really need to address biofilms and the problems caused by them.
We want sustainable therapies for the treatment of infections. What challenges do we still need to overcome to achieve this? And do we have any idea of how we can avoid bacteria from building up tolerance to antimicrobial peptides?
Well, I think the two main issues for applying them as drugs are, the first one is toxicity, which is really outside my area of expertise, but it is a problem that has been holding back the development of AMPs as drugs. I mean, by the way, they attack bacteria, it's basically because they're cationic, so attracted to the charge of the cell surface, and the cell surface charge is completely different with eukaryotic cells. One could assume there is no toxicity, but in the literature you find a lot of indication of toxicity, and that's the main problem. But aside from that problem, so if you have topical applications, that would be less of an issue. And most current developments of AMPs as drugs focus on topical applications. But then again, you get the problem of resistance evolution. Now, in resistance evolution, obviously, at the face of it is very simple. You have a drug, and the drug has a high concentration, bacteria exposed, drives resistance evolution. But obviously, that is slightly more complex. So factors that come into place is, first thing is the population size of the bacteria under selection. Second effect is the so-called mutant supply. So where do they get resistance mutations from? Are there pre-existing mutants in the population that I treat, or in the population of bacteria that infects the host that applies antimicrobial peptides as an immune defense? Do we have horizontal gene transfer? All these questions are important to estimate the risk of resistance evolution. We know that, for example, some antibiotics increase mutation rate in bacteria. And we've previously shown that that doesn't seem to be the case for probably most antimicrobial peptides. And the reason is that because they kill so quickly. So antimicrobial peptides kill bacterial cells within minutes usually, whilst antibiotics usually take hours to kill bacteria. And so antibiotics elicit, not all of them, but quite a number of them elicit stress responses in the cells. And these stress responses lead to the use of alternative polymerases to repair the DNA damage. And these polymerases are not as precise as would if you had a normal kind of duplication of your DNA. So you get error, they're error prone. And the antimicrobial peptide attack doesn't elicit these responses. And that's kind of beneficial. So, and I think the other important aspect are the so-called pharmacodynamics, so the relationship between bacterial growth and concentration of a drug. And an important part here is the so-called mutant selection window. So that's the kind of range of concentration where a resistant mutant of a bacterium does better than the wild type. So that's a kind of drug concentration where we select actively for resistance. And that's mutant selection window is much more narrow for most antimicrobial peptides than it is for antibiotics. And that probably has to do with the killing mechanism and the speed of killing. But basically the concentration of where you have no effect on a bacterial population and where you have a maximum effect, it's a very small distance. For many antibiotics, it's quite a range of concentrations under which you have that, killing before you have killed everybody. And that, we combined that in a theoretical model together with colleagues from the ETH Zurich, with Roland Rieger's lab. And if you can combine these effects on mutation rates and population size and pharmacodynamics, you actually find that the probability of resistance to evolve against antimicrobial peptides is much, much lower than against antibiotics. And now their experimental work and papers that support that. So from the wild, i.e. from medical use, there's only one example, and that's colistin. So colistin is an antimicrobial peptide of bacterial origin. So most antimicrobial peptides are actually, or which are researched and people talk about in the literature, are from eukaryotes and often from animals or plants or fungi. But anyway, colistin was introduced into the clinic in 1960, and the first major reports on resistance against colistin were in 2015. So it was a very well-known paper where they discovered resistance in a pig farm in China, I believe published in the Lancet Infectious Diseases. If you compare that with reports for other antibiotics, where it takes on average 2 to 3 years between the introduction of an antibiotic to clinical usage and reports on resistance, then that's kind of consistent with our modeling efforts and the prediction that resistance evolution has a much lower probability against antimicrobial peptides than it does against antibiotics.
You write in a recent article that if AMPs are to be used clinically, it's crucial to understand their natural biology in order to lessen the risk of collateral harm and avoid the crisis of resistance now facing conventional antibiotics. Have we made progress in understanding AMPs' natural biology better, or what needs to happen next for us to advance further?
You know, when we wrote that sentence, that was only about, a bit more than 2 years ago, and therefore not that much has happened, but we hope we have motivated people to look into that question. I think the first main observation is that when we use antibiotic drugs, with a few exceptions such as diseases like tuberculosis, we usually use a single drug. When we study antimicrobial peptide responses, let's say in an insect, in a mammal, in a plant, upon infection, they release a multitude of different antimicrobial peptides. So in natural situations, antimicrobial peptides, with very few exceptions, almost always act in cocktails. So I think that's a very important thing to keep in mind, and we can go into a bit more depth with that in a minute. The other thing that is important is that when we measure activity in vitro, and the same applies to antibiotics, we do that in standardized solutions, to have standardized measurements of efficacy and so forth. But that doesn't really at all mimic what happens in the context, in the physiological context of an animal or of a patient which we treat with a drug. So the examples also for antibiotics, which have no activity against particular bacteria in vitro, but show activity in vivo. And the same is true for antimicrobial peptides. And if we want to know their efficacy in terms of killing, their toxicity and so forth, we need to be much better, I think, in mimicking the natural conditions. And that not only includes stuff like pH and so forth, but should also include, to some extent, the interactions with other immune effectors. I think that will be very important as well. And only then, I think, we can make more progress on understanding what's going on in terms of the natural situation. Coming back to your question, that will also make it much easier to understand resistance evolution and the probability thereof. Because as I said previously, with an antibiotic, it takes a number of years for us to observe resistance in patient isolates, for example. If you do the same experiment in a lab, against many, probably all antibiotics, it takes a few weeks in the lab to get resistant bacteria. It might be very hard for some, and you need to tinker a bit with the conditions, but bacteria in the end will succeed, and not within years, but within weeks. And that shows really that the natural situation, so the kind of selective arena for resistance in a host, is much more complex and much harder to understand. And now that we are at the cusp of using antimicrobial peptides as new antibiotic drugs, I think it would be very important to have a better handle on these issues. Because otherwise, you can make the safe prediction that if you use an AMP, produces en masse, and use it as an antibiotic, it will be a bit slower, but ultimately we have a huge problem with resistance evolution. And that obviously is something we would like to avoid if we have a new opportunity by having basically a new class of antibiotic drugs.
Research on antimicrobial peptides is progressing. Which aspects are you currently most excited about, and which areas are you personally keen to explore further?
Well, for me, one of the most exciting parts is something I alluded to briefly before, that's understanding the nature of interactions between AMPs. There are some beautiful studies where people have studied the biochemical and biophysical interactions between pairs of antimicrobial peptides. But what I'm personally very interested in is so-called higher order interactions, such as those that we find in an immune response. How do 5, 10 antimicrobial peptides interact? And that's very tricky to tackle. So if you think about it, they have just three, and A and B are synergistic, B and C are antagonistic, and A and B are additive. So what's the overall outcome? That's a very difficult question. Same applies for antibiotics, and there are competing models around and some nice high throughput experimental evolution studies, but there's no clear picture yet of, the interactions are of nature, additivity or whatever, and we can therefore predict higher synergy, lower synergy. I mean, we've done with antimicrobial peptides from the shelf, we've done a study where we did, all two-way and all three-way interactions, and we find that most two-way interactions are actually synergistic, even though we have different antimicrobial peptide classes, and they were from completely different organisms. And the three-way interactions have even more synergy than the two-way interactions. There's also a paper by Pamela Yee's lab where they studied interactions of up to 6 antibiotics, and they find the more they put in the mix, the higher is the likelihood of finding synergistic interactions. But I think that's very challenging in terms of developing theory, developing experimental approaches, but I think it will generate a lot of insight for both really pure science, because it will give us an insight of why natural selection always has come up with cocktails of antimicrobial peptides, rather than an organism investing all its energy into producing one highly potent antimicrobial peptide. And the same is true for that recommendation that we should use new antibiotics or new antimicrobials only in combinations. I can believe that that's an interesting proposition, but we know so little about that might be a bit risky. So I think the interaction question, I think, is one of the most exciting ones, at least for me, and that will hopefully keep me busy for a few years.
Professor Rolff, thank you for all the answers to these questions. There is still a lot to discover. Best of luck for your further research. Thank you.
Thank you very much.